Okay, so today we're starting off, continuing on with muscle stuff. So today and then Wednesday will be on muscle, and then we'll dive into the beginning of the nervous system on Friday. Okay, so today we'll talk about something that we're talking about in terms of muscle, but all cells create ATP in one way or another, right? And that's really what we're talking about. We're talking about energy systems. Okay, but let's start out with this one here. Go ahead and just talk to the people on your team about this one. And make sure you're on the same page and send it in when you're ready. All right, it's maybe about 10 more seconds. Probably need about 8 or so more of you to send it in. Okay. All right, so about 2/3 of you are saying C there, so to turn ADP into ATP, and that really is the primary goal. Some other things happen there along the way. All of those other, well, at least A, B, and D also happen in terms of this, and then definitely we need to make proteins, but it's kind of a different process there that we've already talked about in protein synthesis. All those other things happen, but really the end goal is to make that ATP, right? Make that energy so we can use it for fuel. Okay. So let's talk about the 3 ways that muscle cells in general do that. Okay. And so there's 3 primary energy systems in muscle cells. Okay. Number 1 is called the creatine phosphate system here. Okay. And this is definitely the fastest of all the systems. Okay. It's not about breaking down a food like carbohydrates, proteins, or fat. And it just makes 1 ATP at a time. Okay. If we were to do an all-out sprint, then we would run out or deplete this energy system in about 10 to 15 seconds. Okay, so running as fast as you can, pedaling as fast as you can on a bike under high resistance would pretty much deplete this system in about 10 to 15 seconds. Okay, so that's our first one. Second one's called the anaerobic pathway, also known as the glycolytic system. Okay, glycolytic system. It's called that because it primarily uses glycogen for the fuel. Okay, glycogen again is that stored form of carbohydrate we see right here. Right, and it's primarily used that, so it's called the glycolytic system. It's the second fastest. Okay, not quite as quick at producing energy as the creatine phosphate system. It uses either glucose or glycogen, but mostly glycogen. This one makes 2 ATP at a time per glucose molecule. Okay. And with an all-out sprint, this one will last somewhere but around 2 minutes in general. Okay, so has a bit more fatigue resistant capabilities than the creatine phosphate system, but doesn't give us quite as much power quickly. Okay, and then lastly, the one that produces the vast majority of all of our ATP in the body is known as the aerobic pathway. This one's definitely the slowest. This is the only one that can also burn fat and protein for us. Right, we can definitely use glucose and glycogen for this as well, but this is where we also use our fats and proteins. 32 ATP per glucose molecule instead of 2, right? So much more efficient than the glycolytic system. And so this is definitely the preferred energy system of the body when it can use this one pretty well, because it's the best use of our fuel source. Right? And this one only really is limited by the amount of fuel that we have, and we don't typically run out of fuel unless we're in some kind of starvation state. Okay. All right, so those are the 3. We're going to dive into each of these in a little bit more detail. So let's talk about the creatine phosphate system first. So creatine phosphate system pretty much takes, sorry, all the arrows got messed up on this one, I'm not sure why, so all those in question marks on this slide, they're supposed to be arrows, typically going to the right, I think they're all going to the right. But the idea here is we have creatine, which is a protein that has a phosphate attached to it. Okay, and what happens is that creatine, with the help of an enzyme called creatine kinase. Okay, so that's the key enzyme in this one, creatine kinase. It helps take that phosphate off of the creatine and then puts it onto a DP, right? Which just means there's two phosphates there instead of three. Diphosphate versus triphosphate, right? And then it makes our ATP for us. Okay, so it's just a single step, right? Using that creatine kinase enzyme, taking that phosphate that's kind of stored there on creatine, puts it onto ADP. Now we got ATP. Okay, so super quick, very fast, high energy production. This is used primarily with like single powerful movements. Okay, so think about like doing a vertical jump, running for like less than 10 seconds, right? Somewhere around 10 seconds, 10-second sprint. Could be just like weightlifting, swinging the back, could be throwing a ball, something like that, where you're doing something that requires a lot of power, a lot of energy production fast, and you're just doing it really quickly. That's kind of what exemplifies the use of this. Okay. All right. Questions on that one? So very, very simple, very quick reaction. Okay. And this one is really the basis for creatine supplementation, right? How many people have done creatine supplementation in the past? Yeah, so it's actually one of the best studied, actually the best studied ergogenic aids that's out there. And there's lots of research on creatine supplementation. And some of you that have done it before, what would you say the main benefits have been for you?
I feel like I like more like energy.
Yeah, it's more like
Sometimes even the strength.
okay, almost sort of like what's that? Yeah. Okay. All right, yeah, and that is the primary thing with this. It's not like an anabolic steroid that like actually causes your muscle cells to feel stronger, but what it does, it gives you more energy to push yourself a little bit harder. Right? So you get a greater stimulus on your muscle, because the greater stimulus you have, then the stronger adaptation your body will have against that stimulus, right? So more stimulus, more adaptation. And that's the idea, right? If you can allow yourself to push yourself a little bit harder because you have more energy to do that, then you can get more adaptation on that. And that's really what creatine supplementation has been shown to do. So, yeah. Other questions on that? Okay. All right. So the next two energy systems, when we're talking about using carbohydrates, they both start with glycolysis. Okay, and you don't need to know all the details of the glycolysis pathway for this class, but what you do need to know is that it starts with glucose and ends with pyruvate, right? That's what glycolysis is taking glucose, breaking it down through that whole series of steps and forming pyruvate. Okay, and so this is really the common pathway of both the glycolytic system and the aerobic system when we're using carbohydrates. Okay, so again, definition of breakdown of glucose to make pyruvic acid, aka pyruvate. Okay, happens in both the aerobic system and the anaerobic pathways. And it can start with glycogen or glucose. It's technically called glycogenolysis when we start with glycogen, but don't worry too much about that for this class. The other things that come out of it are two ATP molecules get formed directly in this part of it. And then we get two NADH molecules, and we'll talk about the importance of those coming up. But those are really the key parts that are going to help us make most of our ATP at the end of the aerobic system. Okay, so glucose, pyruvate, that is glycolysis. Okay, so then once we get to pyruvate, pyruvate really has two different options for us to go through in humans at least. It has the aerobic pathway and the anaerobic pathway. If it goes the aerobic pathway, it makes acetyl-CoA, right? And acetyl-CoA is what's going to start the Krebs cycle for us. Okay, if it goes the anaerobic pathway, it's going to turn into lactate, and that's the end. That's it. That's the end of the glycolytic system. That's the end of the anaerobic pathway. Okay. When we're talking about other microorganisms like funguses and fungi and certain bacteria, they have a third option for this, which is what turns it into alcohol, right? Which is what how they make wine and beer and things like that, and put yeast into something that has a bunch of sugar in it, right? Like a fruit of some sort, like grapes for wine, right? Or barley and hops, or barley and wheat for beer, right? It takes that carbohydrate and turns it into ethanol, right? And that's how alcohol is formed. Humans can't do this pathway. Okay, so this is kind of the three different ways that pyruvate can go, but in humans, it's only these two ways. Okay, so let's talk about the anaerobic pathway a little bit more. Okay, so glucose to pyruvate again is glycolysis. No oxygen is used in this pathway. Okay, one thing to note is when we get down to pyruvate, we make two pyruvates. Okay, so one glucose makes two pyruvates. Okay, which then will turn into two lactic acids. Okay, and so then what the other stuff we get out of that, like I already said, is we get two ATP molecules there. And then we get some NADH. We'll again talk more about what that one does for us soon. But again, this is really fast, high energy production. Can get us energy, can get us ATP really quickly, but it's also a terribly inefficient use of our fuel. Right? If we're only getting 2 ATP out of it, we could be getting 32 out of it if we did the oxidative pathway, then that's not something we want to do very often, right? But our body will resort to this when it needs ATP rapidly. Okay, when we're trying to sprint, we're trying to do something very fast that it's just faster than our aerobic system can keep up with, then it'll use this pathway instead. But the cool part about this pathway is that it's self-limiting. Okay, so the more we use this pathway, the more our pH drops, because we build up hydrogen ions from that lactic acid, and that starts burning. So we physically feel that, right? That feels like pain. But it also, all those hydrogen ions also go and bind to all the enzymes that drive all of these reactions too, right? So there's every single one of those reactions is driven by proteins that are helping drive those reactions. And when a bunch of hydrogen ions start binding to those proteins, they start changing shape. They start not doing their job very well. And so that slows down our ability to use this pathway, right? So it's negative feedback that this pathway gives to itself, right? It doesn't allow us to do too much of this pathway, or else it just slows us down. Okay. And so that's a key piece to this pathway. Okay. So where in the cells does this occur? This is happening right out in the cytosol of our cell, so not in the mitochondria, but just kind of out in the open, in the fluid part of our cell. Okay, but then at the end, again, we're making this lactic acid. What happens when that lactic acid gets released? Well, it releases its hydrogen ion. Okay, it's an acid, a weak acid, but it's an acid, so it's going to release that hydrogen ion. Okay, and then lactate used to be blamed for all kinds of bad things like muscle soreness and all kinds of things, injuries, but really what we know is that now is that lactate is actually so close to being pyruvate, which is a really important fuel, that for the most part we reuse that lactate. Okay, so one of the things we can do with it is that if it gets into our blood and then circulates back into our liver, then our liver can turn it back into glucose. Okay, that takes some energy to do that. It's called the Cori cycle. But it can do that. Okay, that's one way to do it. But the more common thing that happens to it is that it gets shuttled right over to another cell that might have more mitochondria than the one that it was produced in, and that cell turns it right back into pyruvate, and then it can go through the oxidative system. Okay, so most of our lactate is just kind of recycled, and we use it as fuel in our more oxidative cells. So that could be in our oxidative muscle cells, it could be in our heart cells, kidneys, or also our brain is a high user of lactate. That's in the every one. So lactate is by no means like this waste product that we got to get rid of. It's more of a fuel that just needs to be converted back into something a little bit more useful, and a lot of different cells can do that for us. All right, but then the hydrogen ion that the lactate releases, this is really what causes the problems with the system, right? Decrease of pH, causes that burning during exercise, but it really is completely back to normal within 30 minutes, usually of exercise, and for sure, it's all gone and back to normal in 2 hours. Okay, so to talk about lactate being related to that delayed onset muscle soreness, we might have 2 to 3 days after a workout just, this has nothing to do with it. Lactate is not responsible for that muscle soreness we might have. Okay. Questions about any of this? Okay, yeah, so go ahead and let's do this one. Go ahead and talk through this one with people on your team and vote when you're ready.
I'm gonna like read the.
All right, maybe another 8 or so more of you. Go ahead and send in your answer. Okay, good. It looks like almost all of you. So yeah, 93% of you are saying C. So whether the aerobic or anaerobic system is used. Absolutely. Okay. All right. Well, we're gonna now dive into the aerobic system next. So this is our most used system. For sure, this makes 99% of the ATP in our body. And yeah, so this is definitely the one that's used the most. And just an overview of it is we still got to start with glycolysis if we're talking about carbohydrate, which is the only one that we'll dive into in detail this class. And then we go from glycolysis to citric acid cycle, also known as the Krebs cycle or the TCA cycle. And then we go to the third step, which is the electron transport chain. And so the oxidative system includes all 3 of these steps together. Okay. And really, there's another little step in between here as well that we'll look at in a second. Okay. So yeah, so if we start with, if we're using carbohydrates, which is what we're talking about in this class, and we'll start with glycolysis. That other little in-between step that I mentioned a second ago is this step, turning pyruvate into acetyl-CoA. That's not part of glycolysis, it's not part of the Krebs cycle. It's just a step in between. Okay. Then we do the Krebs cycle, then we do the electron transport chain, all 4 of the oxidative phosphorylation. Okay, so the products, byproducts, ATP is the primary thing we get out of this. Okay, so in total, we're gonna go with the number of 32 for this. Okay, this number varies quite a bit when you look at different textbooks and different sources. It used to be a lot higher than this, and a lot of the biology textbooks still show the number of like 39 or 38. And a lot of that has to do with just kind of how efficient human cells are, and it's probably lower than that. And so we're just kind of choosing a number to go with for this that comes from the latest stuff that I've seen, which is 32, and we'll talk about that a little bit more. So if you had a different number from another class, just go with this one for this class. Okay, okay, this is where our CO2 comes from as well. Right, so the CO2 that we breathe out, it comes from this aerobic system, and we make a bunch of water doing this too. We'll see where we do that here in a sec. Okay, so substrates that can be used for this system: glucose, those are the carbohydrates; free fatty acids, those are our lipids, right? And then amino acids from proteins, right? We can break down all of these different macromolecules in this aerobic system. But again, we're only diving into the glucose one in detail right now. Okay, so this kind of gives us an overview of what this looks like, right? So we're going here, carbohydrates, glucose, turn it into pyruvate, acetyl-CoA, and then we go to the Krebs cycle, electron transport chain, okay? If we were going to talk about the lipids one, then we go through a process called beta oxidation. And beta oxidation is eventually going to make a bunch of these guys for us again, and then go to the electron transport chain. So you'll see that all of these use the electron transport chain and the Krebs cycle in some way or another. Proteins will break down into individual amino acids, and then they all jump into this cycle, into the different, some turn into pyruvate, some turn into acetyl-CoA, some turn into different parts of the Krebs cycle. Okay, but overall, we're still going to use the Krebs cycle and the electron transport chain for all forms of fuel that we're breaking down. But again, we're really only going to talk about this one in detail. Okay, so here's the Krebs cycle. Again, not something you need to memorize for this class, okay? But I do want you to kind of understand what goes into it, the primary thing that goes into it, which is acetyl-CoA, and then what important products we get out of it. Okay, and then the other thing to know is that with a single glucose, we're sending 2 pyruvates down and 2 acetyl-CoAs down. So that means we get 2 full turns of the Krebs cycle with each of those. Okay, so the Krebs cycle portion here, okay, also known as the citric acid or the TCA cycle, always going to start with acetyl-CoA. Okay, and then each glucose gives us 2 of those acetyl-CoAs, so we get 2 turns of the cycle. Okay, so the products we get out of this, we get some NADH, some FADH2, and then we get 2 ATPs for the 2 cycles. Okay, the NADH and the FADH2, well, really the NAD plus and then FAD, these are both derived from vitamins that we need to eat. So vitamin B3 and vitamin B2, riboflavin and niacin. And so these are very important essential vitamins for us that help make up the enzyme complex that carries these electron-rich hydrogens over to the electron transport chain. Right. So the primary goal and the primary reason these things are so important is because they take electrons from earlier steps in the breakdown of these food products and drop them off of the electron transport chain. And without those electrons, we can't make ATP. Okay, so it's crucial that we're making these here in the Krebs cycle, and we also made a couple of NADHs up in glycolysis as well. Okay, so let's talk about that next step then, the Krebs cycle, or sorry, the electron transport chain. This is the very last step in the aerobic pathway. Okay, it uses a type of transport called secondary active transport, right? Again, if it's active transport, what does that mean as far as which way we're moving something in regards to its gradient? Again, absolutely. Okay. So it uses secondary active transport. So that when we use secondary active transport, that means that one thing's moving against its gradient, and something else is moving with its gradient, right? And we're using the energy from that thing that's moving with its gradient to drive something else against its gradient. Okay. So in this case, the thing that's moving with its gradient is the electrons. Okay. And the thing that's moving against its gradient is hydrogen ions. Okay. And so by using those electrons, we're pushing hydrogen ions against their gradient, which is going to build up this big gradient of hydrogen ions that we're going to use to make ATP. So we're going to use that hydrogen ion gradient to make ATP. And we'll watch this little video here in a sec, and then I'll explain it a little bit more as well. But we're using this hydrogen ion gradient that we created with secondary active transport to then drive the formation of ATP. Okay. And then oxygen, this is where oxygen comes into play. The whole reason that we breathe, right, is that oxygen can be the final acceptor of those electrons at the very end of this chain. And with all of this, this is how we get 28 of those 32 ATP are created in this little last step here. So this is where the vast majority of all ATP is made. Okay, let's hop over to this little video and we'll watch the steps in there, and then we'll come back together. What's up? There. Right there. Thank you.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria, so cells can use it. Mitochondria consist of 2 membranes, an outer membrane, separating it from the cytosol, and an inner membrane, surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
I find brilliant gifts at songs.
Yeah, so let's let me kind of walk us through some of the steps of that here with this image. And I think the best analogy for understanding how this thing works is thinking about what's called a hydroelectric power plant. How many people have toured something like Hoover Dam or a dam like that? You guys been on one of those tours? Anybody? Raise your hand. Yeah. Okay, so you okay. And so if you've been there, Hoover Dam is the one that I've toured. And when it was built, it was the largest in the world. It's not the largest anymore, but it's about 700 feet tall. Hoover Dam is, right? And Lake Mead is right there. That's the water that's being held in. And the way that the power plant within the dam works is that water it gets let in the top of the dam, right? And then it goes down these big pipes and it falls 500 to 600 feet, right? So water is falling down 500 feet. And there's a ton of energy in water falling, right? Anything moving, there's a bunch of energy. And what it is, it falls down, it hits these big turbines and spins those things around. Some of you remember this part, yeah, in the tour. They always make a big deal out of it. They love using the word dam, like during the tour, like, oh, you want to take the dam tour? Oh, this dam tour almost over, stuff like that. They like joking around about that when you're doing the tour, but so water falls that far and it spins this big turbine. This big electromagnetic thing, somehow that makes electricity. I'm not 100% sure how all that works, but the fact that mechanical energy is turning something that can be converted into electrical energy, okay? And this is very similar to that, okay? What we're doing is we're taking all of these hydrogens up here, we're pushing them all there with the power of the electrons, right? So the electrons get dropped off here by NADH and FADH2. And since we have oxygen down here soaking up all these electrons, there's not many electrons down there on the far right side here. Okay, so there's a lot of electrons on the left side, not many on the right side. So that means there's a strong gradient for these electrons to go this direction. Okay, and so there's power in that gradient, and the power in that gradient is used to push the hydrogen ions against their gradient. That's how we can build up so many of them up here. I think of that kind of like a catapult, right? If you've watched any of these kind of period piece movies or shows like Game of Thrones or anything like that, where you got like this catapult, right? There's like an empty side of the catapult here. It's like a big teeter-totter, you know, the empty one, and then one with a rock on the other side, right? If they drop a big rock on the empty side, it can fling the other one across. That's pretty much exactly how secondary active transport works, right? The electron is the one that we're dropping from high, and it kind of it turns this protein in such a way where it grabs onto that hydrogen ion and throws it across the membrane, right? And so that's how we get all these hydrogen ions built up there. And now these hydrogen ions are just like the water falling down the dam, right? There's so many of them up there, they want to shoot back across because it's more negatively charged on the other side, and there's not a lot of hydrogen ions over there. So they rush back across just like the water falling down the dam. And when they do that, they spin this big complex called ATP synthase, and that's what makes all their ATP for. So it's the spinning of this big rotor in ATP synthase that just takes a phosphate, pushes it onto ADP, and that's how we make ATP. And the charge difference across the membrane, you know, you kind of think of this like, oh, it's like little hydrogen ions, like whatever, like shooting across, not a big deal. If you were to extrapolate the charge difference across the membrane from one side to the other to like a like size version of this, so that a membrane is about 1/1200th of the width of a piece of paper. Okay, so if we were to like extrapolate that out to life size, the charge difference across the membrane would be about the same as a bolt of lightning, about 30,000 volts of energy across the membrane. And so this is extremely powerful complex that is doing this. And so when that hydrogen rushes across, it makes the ATP for us. Okay. And the last key step that's really important down here is that, again, oxygen is the final electron acceptor. Okay. And so we're seeing this equation down here at the bottom. I'm going to erase some of those things. There's too much drawing down there. There we go. So we see hydrogen ions coming together with electrons and oxygen to make water, right? This is what's called metabolic water production. But those two electrons coming into that are showing that oxygen is the final electron acceptor. So that almost seems like a minor role in all of this, right? But if we didn't have oxygen there to accept those electrons, and all of a sudden we would start building up a lot of these electrons down here, that would knock out our gradient for electrons, right? If we don't have a gradient for electrons, we can't push the hydrogen ions across the membrane. We don't lose that power to do secondary active transport. So we can't make ATP, right? And we know that if we don't have oxygen for more than a couple minutes, we're done, right? And so just the fact that the oxygen's there catching those electrons and bringing them out, maintaining that electron gradient is absolutely crucial for us for that. So that's kind of the basis of this electron transport chain and oxidation phosphorylation. Questions on that? Okay. All right, so take a sec just with the people around you and just kind of backtrack and fill in how many ATP we created at each of these little steps here. And then on your clickers, you can send in that total number again, just to make sure we're all on the same page.
Oh, that's two.
All right, so you're sending in the number there of the total ATP. You can start talking about the next two questions with your team if you want while you're waiting, because there's two more clicker questions coming up. All right, make sure to hit send with this type of question. Maybe 3 or 4 more of you. Just everybody go ahead and hit send so you get it in there. There we go. All right, so glycolysis, how many do we make in glycolysis? 2, Krebs cycle, 2, electron transport chain, 28, total 32. That's what most of you said. Good. Okay, so we got that. Go ahead and talk about this one, and I'll change it so you can send a letter in. And go ahead whenever you're ready.
Yeah.
I thought it was
All right, couple more people send it in. Almost got everybody, looks like. Good. So the vast majority of you, 98% of you are saying C, to accept electrons at the end of the electron transport chain. Perfect. Okay. And then let's do this last one. Sorry, again, those are arrows going to the right. What's the correct order of events there when we're using glucose? All right, maybe 5 more seconds. Need a few more of you to send it in still. There we go. Good. All right, and 93% of you are saying D there. So we start with glycolysis. That's glucose to pyruvate. Then we got to turn pyruvate into acetyl-CoA. That's going to start the Krebs cycle. Krebs cycle is going to make most of those NADHs and FADH2s. Those are going to go the electron transport chain to finish us off there. Okay, good. And then let's take the last couple minutes here and just kind of fill in this little recap of the different energy systems we just talked about. Okay, go ahead, talk through this with the people on the team, fill it in. Come back together here in just a couple minutes.
Is it anaerobic or aerobic flows? Not very long. Also doesn't produce much.
That was two.
Oh, yeah.
Oh.
That one's just like policy. So. As long as you're
All right, let's walk through this briefly together here. Okay, so as far as let's do speed and endurance first. Okay, so creatine phosphate system, as far as speed goes, where are we at there? Yeah, this is our fastest, right? Anaerobic pathway is kind of in the middle there, right? And then aerobic pathway would be the slowest. Okay, then as far as endurance goes, creatine phosphate system got the least, right? Again, this one's middle. And this one is most endurance. Okay. Number of ATP produced, creatine phosphate system. Yeah, it's just one at a time. Anaerobic pathway, 2. Aerobic, 32. Okay, and then substrate required. For the aerobic pathway, let's start down there. What can we burn with this pathway? Glucose. What else? Lipids and proteins as well. Okay. We also need oxygen for this one. Okay, so that'll be another required substrate. Anaerobic pathway. What do we need to do this one? Glucose or glycogen, and that's more common that we're using glycogen. Okay, and then creatine phosphate. We need creatine. That's about it, but we don't need any kind of food product, as in macromolecule creatine. We do eat creatine in muscle, but we don't need glycogen. We don't need carbohydrates, lipids, or protein. All right, guys, we got it. Have a great rest of your day. We'll see you on Wednesday, Doctor. Okay.